Hello! Today we're going to continue talking about the structure of muscles, but we're going to get smaller instead of bigger today. So yesterday the smallest thing we talked about was a myofibril. That was like that single tube, like a straw, and we talked about how it contains sarcomeres. The sarcomere is the unit that actually contracts. So today I want to draw a sarcomere so we can start talking about how a muscle is able to contract. So let's start with our boundaries. The middle of the sarcomere is called the M line, so that's not too bad. M is for middle. And then coming out here on the other side is a Z disc. This is like one edge of the sarcomere. And then coming over here on the other side is a Z disc. So right now we've drawn one sarcomere. If they would continue off in both directions, but let's just draw one so we try not to make it like super confusing to start off with. Okay, so anchored to the M line is a myofilament called myosin. So that red thing that I just drew is supposed to be myosin. And myosin is known as the thick filament, so we can try to make ours look a little bit thicker just so we can distinguish it there. And we can go ahead and draw some more. Myosin, myosin. Another important myofilament is actin. Actin attaches to the Z disc and then heads out like this. So it kind of overlaps the myosin a little bit. And actin is known as the thin filament. So myosin, thick, actin, thin. Okay, this is looking pretty good. Another thing, we need to actually connect the myosin to the Z disc so it's not just kind of like floating in space. Because if we want our muscle to contract, it needs to have something to attach to. So there is another myofilament called titin that looks like a little spring. That's why I'm trying to draw it squiggly like that. And it's going to allow the actin, like the whole sarcomere, to basically move. So we have a spring inside of our sarcomeres that's holding it all together. So the myosin attaches to the M line and the Z disc. That will be important for us later. And yeah, there we have it, sarcomere. In reality, it would continue, so I could have drawn the actin, I could have drawn the actin still coming out in this other direction, and then over here there would be another M line, and that M line is going to have myosin coming off of it. But try not to confuse us too much for just starting off. For a long time, people thought that when a muscle contracted, it was literally getting shorter. So this idea that there's these little myofilaments like actin and myosin that are able to slide past each other, that was a pretty revolutionary, controversial idea. Here we go, the sarcomere that we just drew. Notice when we look closer at the myosin, it has little heads sticking off of it. The heads are only present near the end. Oh, and I want to label a few regions on this sarcomere, so you should go ahead and label the part that we did together. So one region is called the A band, and it is the same length as the myosin. So I'm just drawing a little thing right there. That is the A band. If we looked at the A band underneath a microscope, the A band would look dark, compared to the actin because the myosin is thick. So the A band is also known as the dark band. And then if we looked at the part where there's only actin going this way, that's called the I band. And this would continue on to the other sarcomere. I'm just only labeling it for us. So one sarcomere is going to contain the entire A band and the I band, and this is known as the light band. And then let's take another look at the middle of the A band. This very center region right here is myosin only. So like most of the A band is actin, myosin, actin, myosin. This little center region right there is only myosin. Because it's only myosin, it's actually going to appear brighter. That is called the H zone. H means bright. Hele means bright. Let's look at this on an actual sarcomere so that you can maybe tell what I'm talking about. So this is an actual image of a sarcomere. So to start by orienting ourselves, this whole region right here is the dark band. So that must be the A band, so that must be where my myosin is. And then this region over here that's light, this is the I band. So this dark line right here, that would be the Z disc. And then over here, that would be my other Z disc. So now I've basically highlighted one sarcomere. And if you look in the middle of the A band, this region right here that looks lighter, that is the H zone. So it's the part of the A band where it's myosin instead of myosin and actin. And finally, here's a closer look at myosin and actin so we can start to get an idea of how they can contract. Myosin has heads on it, and the heads have a binding site on the end where they're able to bind to actin. So when a muscle contracts, the myosin literally reaches out its head like an arm, and it like grabs the actin and pulls. It also has a spot where it can bind ATP. ATP is the energy for the muscle contraction. And then we drew actin like this, but notice in reality it's made of like all these little teeny subunits, like little beads, and they have a hole in them, which is a binding site for actin. So like this myosin right here would bind to this part on the actin. A couple features that we didn't draw on our other actin is that actin has another protein going around it called tropomyosin. If we look at the tropomyosin, what does it look like it's doing? It's going right here and it's blocking all of those binding sites. So tropomyosin is able to block the binding sites and basically prevent the muscle from contracting. And tropomyosin is held in place by a little protein called troponin. So what we're going to find is that this allows us to control when a muscle contracts. We can tell troponin to move, and it will pull tropomyosin out of the way, and then it will free the binding site so the muscle can contract. So they're not always on or they're not always off. They can be controlled and turned on as we need them. And that's a wrap. Please email me your questions.